So now we are going to start the next session, and we all need to know hepatitis E virus updates. So I would like to invite Professor Dr. M Sultan Khuroo.
I shall narrate some extraordinary events while uncovering this remarkable human interest story. On this auspicious occasion, I wish to pay tribute and homage to my parents and family who educated me, taught me dedication, patience, empathy, and stood behind me through thick and thin. For without that, I could not have conceived, worked, and walked on the path of patient care, teaching, and research in medicine and biology. The discovery of hepatitis C is a remarkable human interest story related to the complexities, the missteps, the near misses, and the ups and downs, as with many other similar events in history. The story is fascinating as it originated from one of the most remote regions of the world with very hard weather conditions and primitive healthcare and investigative facilities. The story also focuses on the fact that discoveries do not necessarily require high-tech laboratories or institutions with cutting-edge research facilities, but can be accomplished in very primitive situations, such as in focus. The discovery of hepatitis E has had a major global impact. Hepatitis C is a remarkable human pathogen that has completed the alphabets of human hepatitis viruses. 1/3 of world population is infected with hepatitis C, with an estimated 14 million symptomatic cases, 300,000 deaths, and 5,200 stillbirths annually. In developing countries, hepatitis E genotype 1 and 2 cause massive waterborne epidemics with high prevalence of endemic disease and selectively kills third trimester pregnant mothers with high percentage of fetal and neonatal deaths. In industrialized countries, hepatitis E genotype 3 and 4 infections are being recognized as important clinical problems spread either through foodborne zoonotic transmission or through HEV infected blood and blood products, and cause chronic hepatitis and cirrhosis in solid organ transplants and also neurological syndromes. In fact, hepatitis E has been designated as one of the most successful foodborne zoonotic infections in human history. Hepatitis C has a remarkable heterogeneity and infects a wide range of animals. A recombinant hepatitis C vaccine, the HEV 239, has been licensed in China, and all of us are looking forward to its availability and applicability. The story of hepatitis C is historically linked to the Gulmer-Kashmir epidemic in 1978. The massive, unprecedented epidemic posed an intriguing puzzle that needed a systematic approach to a solution, belief in oneself, and conviction to explore the unknown. Otherwise, there were numerous hurdles to cross for a young physician like me who had just completed his postdoctoral training. These included hard weather conditions, primitive healthcare structure, time factor to investigate a short-lasting massive epidemic, immediate need for a large survey team, availability of funding, and personal health risks. To face these challenges, I opted to live in the area and strengthen the healthcare and offer care at doorstep. None of my physician colleagues agreed to join me, and today I tell them, thank you for not joining me so that I could do it alone. 500 healthcare workers joined me, needed a 2 weeks of in-service training, and formed an efficient survey team. I made immediate funding available generated from my practice, and the project received no external funding. With all this, we planned an ingenious door-to-door survey protocol for the entire epidemic area to identify each next case of hepatitis over an extended period. This slide depicts the events of the epidemic over the period. The epidemic was announced on November 2, 1978, and we were there within 2 weeks. 4 door-to-door surveys done at 4 to 6 week intervals were conducted in 200 villages over a population of 600,000. The epidemic curve was highly compressed and lasted for 9 weeks with no secondary cases, with a weekly occurrence of up to 4,000 icteric cases, amounting to a total of 20,000 icteric cases with 600 deaths, a phenomenal occurrence. Based on epidemiological, clinical, and histological and serological data from this innovative study protocol of the epidemic, we announced the existence of a new disease and called it epidemic non-A non-B hepatitis, and postulated the existence of another human hepatitis virus hitherto not known. The features of the epidemic non-A non-B described by us in detail in this slide became a benchmark for studying hundreds of epidemics of hepatitis C in developing countries over the next 4 decades. We used 3 innovative ways to break the news of the discovery of the new disease. An award-winning culinary oral presentation was given at the annual conference of the Indian Society of Gastroenterology held at Pune on October 12, 1979. Over a short period of 6 months, the American Journal of Medicine, the Green Journal, carried 2 full articles on the disease and published them in June 1980 and January 1981 issues, with 2 more communications in Lancet. With the personal help and intervention of Nobel laureate Dr. Blumberg, I had the distinction of giving 3-slide, 3-minute plenary presentation to the first international conference on viral hepatitis held at New York on March 30, 1981. All this led to a buzz and started a series of inquiries, support, and collaborations. However, the existence of the epidemic non-A non-B as a new disease entity did not go well with the skeptics, who were convinced that this was a classical example of waterborne hepatitis A and denounced my data based on an opinion that the hepatitis A testing, which I had put forward, was faulty. To counter the A versus non-A controversy, I shipped thousands of sera to many reputed laboratories of the world to confirm my findings. To look for a new agent, I collaborated with Robert Purcell at NIH. Kashmir samples reached his lab on August 11, 1981, in an excellent condition for animal transmission studies. On January 1, 1982, Bob Purcell wrote to me, quote, "Muhammad, because of shortage of chimpanzees, we have not been able to inoculate your specimens to date, but they will be inoculated as animals become available this month. Bob Purcell." The unfortunate lost opportunity. While I was struggling to find the animals for transmission and did find them later at the ICGB, New Delhi, late Dr. Balian from Russian Academy of Medical Sciences in Moscow, Russia, showed extreme zest to explore the unknown. Through a self-experimentation, he identified virus-like particles in his stool samples, and this became the first stepping stone towards finding the etiological agent of epidemic non-A non-B hepatitis. We experienced a major setback in hepatitis C research from 1984 to 1991 as infectious samples from humans contained few virus-like particles, not enough for cloning and sequencing. This frustration ended when Dr. Bradley at the CDC found large quantities of virus-like particles in the bile samples of experimentally infected macaques. Soon, Dr. Reyes from Gene Lab on the West Coast partially cloned 1.3 kilobase of the virus, which led to the subsequent cloning and sequencing of a full-length 7.6 kilobase virus and defining the genomic structure. Now we have a meticulous details of hepatitis C viron, the hepatitis C virus genome with its 3 open-reading frames and their expression, and the structure of the virus-like particle with the detailed structure of the capsid proteins with their 3 linear domains, namely the S, the P1, and the P2 domain. From 1978 to 2013, we encountered 10 periodic epidemics of hepatitis C in Kashmir and thousands of cases emanating from endemic disease. We employed innovative study protocols to understand the disease and formulated several concepts to explain its intriguing nature. In return, we described 5 hepatitis C virus disease forms, studied unique relationship of hepatitis E with pregnancy, and established 4 distinct routes of transmission of hepatitis E virus infection. We established that hepatitis E virus antibodies are long-lasting and gave a concept of cohort phenomena to explain the periodicity of epidemics of hepatitis E. In addition, we established peculiar hepatic histological features of hepatitis E virus infection under the term of cholestatic hepatitis with bile plugs and rosette formation. Why children do not manifest with hepatitis E and that hepatitis C infection is self-limiting disease were studied in depth and explained. All these data were published in over 60 scientific publications in high-ranking journals over the years, and lately were reviewed by me in 4 review articles. In addition to the above, we in a seminal paper were the first to show that hepatitis C virus infection is the only hepatitis virus that gets transmitted uniformly to the fetus, causing severe fetal and neonatal disease. Based on this, we have proposed the concept of mirror syndrome as a possible cause of severe maternal disease caused by hepatitis E. Recently, we have made a major inputs on the understanding of the pathogens of hepatitis C in pregnancy and unraveled the unholy alliance between the pregnancy and hepatitis C virus infection, which was restricted to hepatitis C genotype 1 and occurs only in developing countries. Studies have confirmed that hepatitis E virus genotype 1 and not other genotypes is charged with ORF4 protein, and this unique protein with unique properties enhances the viral replication in the liver and enhances liver damage. It infects and causes significant pathological changes to the maternal-fetal interface, which loads maternal blood with cytokines and breaks the maternal-fetal barrier to cause vertical transmission. Vertical transmission causes severe fetal infection and possible release of toxins into maternal blood, triggering severe liver disease and DIC. On the other side, enhanced hormonal alterations and immune dysregulation of white nature also promote viral replication with a negative maternal outcome. The role of hepatitis E virus mutations and high viral load may also contribute to poor maternal outcomes. Hepatitis C was for long nicknamed as a disease of those third world countries. However, hepatitis C has moved beyond such borders with the discovery of swine hepatitis C virus by Dr. Meng from the Maryland College of Veterinary Medicine at Virginia and working as a visiting scientist with Bob Peirce's lab at NIH. This opened a huge Pandora box of hepatitis C virus infection in the animal kingdom, and at present, viruses from large number of animals have been well characterized, cloned, and sequenced as seen in this slide. In view of the marked heterogeneity, the International Committee of Taxonomy of Viruses has proposed a taxonomy, history, and classification of hepatitis C virus as shown in this table. Hepatitis C virus is listed in one of the distinct families of Hepiviridae with 2 genera, 5 species, and a number of genotypes. The new classification has reduced confusion in classifying the existing isolates as well as the new hepatitis C isolates sequenced from humans as well as animals. Hepatitis C genotype 1 to 4 have distinctive geographical predilection with 4 distinctive epidemiological zones, namely the epidemic infested hyperendemic zone, the high endemic zone with no recorded epidemics, distinctive zone restricted to Egypt, and the industrialized countries with sporadic autochthonous infections. While industrial countries have controlled the spread of hepatitis C by ensuring clean potable water, genotype 3 and 4 spread through ingenious foodborne transmission. Domestic pigs, wild boar, and sika deer show cross-transmission of hepatitis E, and consuming raw and undercooked meat or liver, a luxury in such countries can cause outbreaks of hepatitis C. More serious is the practice of visiting supermarkets and buying raw liver or figuratively sausages and eating it undercooked or raw. Significant percentage of such livers and sausages contain live virus. Sewage from pigs in such countries can flow to waterways, and visiting sea beaches and consuming infected mollusks have led to small outbreaks of hepatitis C virus infection. Hepatitis C was considered an agent responsible for acute hepatitis that does not progress to chronic hepatitis. However, Kamar et al. Reported on 8 out of 14 patients of hepatitis C genotype 3 in solid organ transplants which progress to chronic hepatitis. Chronic hepatitis E is reported in patients with solid organ transplants, human immunodeficiency virus, and hematological malignancies. HEV infection is being recognized as an important clinical problem in many industrialized countries under the several scenarios. Zoonotic foodborne transmission can lead to autochthonous outbreaks or sporadic cases of hepatitis E. Recently, data about such infections have been reported from several countries. Over 21,000 such infections with 37 outbreaks and 28 deaths occurred in European Union countries from 2005 to 2015. Transmission-transmitted hepatitis C is becoming a problem in many countries and has led to protocol of screening of donors for HEV in many such countries. It is now well accepted that HEV genotype 3 infection can induce chronic hepatitis and cirrhosis, solid organ transplant, patients with HIV and those with hematological disease. The clinical profile and follow-up large cohort of such patients has been reported and predictors of chronicity identified. Lastly, hepatitis C can cause a number of extrahepatic manifestations of these. Neurological syndrome is well studied by Dr. Dalton from UK. The news that rat hepatitis C virus infecting humans in Hong Kong has thrown shockwaves throughout the world community and is a wake-up call for the need of a clean environment that is free of rodents. While rats are known to transmit several other diseases in humans, including plague, Lassa fever, and leptospirosis, this is the first report, case report in humans of a rat hepatitis E virus in the humans. Hepatitis E vaccine has had a major breakthrough in hepatitis E research. HEV 239 is a particulate vaccine expressed in E. coli and consisting of ORF2 from a hepatitis E genotype 1 Chinese strain. This vaccine has successfully completed phase 2 and phase 3 trials in China and is commercially available in China as Hecolin in 30 microgram doses to be administered at 0, 1, and 6-month regimen. At 4 and 5 years, vaccine efficacy has been 86.8 and 87% of the vaccine group maintained antibodies and 9% controls developed only antibodies. Let us summarize what are our challenges in the control and cure of this global human infection. We need a better and more accurate diagnostic tool which should be available and extendable to the regional world with primitive healthcare facilities and even those with politically unstable and disturbed regions. We need to get a better understanding of many perplexing issues about the epidemiology of hepatitis E, both in those developing and industrialized world. We need to develop treatment strategies for hepatitis C virus infection and management plan for those with severe infections, especially those with liver failure. Finally, we need to develop and implement effective preventive strategies, especially related to hepatitis E vaccine. Ladies and gentlemen, it has been a pleasure to present to you the tale of hepatitis C for the last 40 years. This started with my extreme belief that epidemics of jaundice and resultant mortality in pregnant women in our community had a hidden saga and to uncover needed hard work, persistence, belief in oneself, and honesty of purpose. It needed courage to stand on its feet to fight the skeptics and listen to the voice. It meant spending days on snow-bound, rubbish roads of those villages and feeling the pain and anguish of the sufferers. It needed nights to spend awake with pen and paper to write one believed is true and to uncover. While this journey was treacherous, hard, and full of failures and some successes, the end has been pleasant and rewarding. For discoveries do not come without a price to be paid. Mr. Chairperson, sir, today I feel honored to be invited to deliver this.
Thank you so much, Professor Dr. Mohammed Sultan Khuroo, for wonderful presentations on hepatitis E virus updates. I'm thank you so much for the chairpersons and the audience for listening carefully. And now I'm going to invite as a moderator Dr. Farhana Kiani. So please come into the stage.